Right, well, good afternoon, everybody. And it's not just a good afternoon because it's Friday, it's because we've got a new DRIIMB webinar for as part of a long running series that's just starting up. So this is the second um, guest lecture in the series. So I think last time we didn't have a website for DRIIMB, so that's new. So I'll post it in the chat in a bit. But so DRIIMB is the new project that's a combined initiative between four CCPs, so it's CCPBioSim, CCP4, CCPEM, and CCPN. And it's all about trying to combine efforts around simulation and experimental software. And in this lecture, we're inviting Jas, who works with me here at Daresbury, but she works on the PSDI, which is the Physical Sciences Data Infrastructure Program to establish a UK national data infrastructure within the physical sciences remit. So Jas is going to give her talk called BioSimDR, which is BioSim Data Resources, and it's a collection of tools and infrastructure specifically for biomolecular simulation. So I'll hand over to Jas now and she can start.
Thanks, James. Okay, so yeah, thank you so much for the introduction. So BioSimDR is what I'll be talking about today. And so this talk is going to go over what biomolecular simulators do, very roughly and succinctly, hopefully, and then the data infrastructure that's currently available for these simulators and where we're lacking, and then how BioSim data resources can help and how our tools and services can support biomolecular simulators. So firstly, for those who may not know or kind of are new to this field of what biomolecular simulators actually do. So we take 3D structures resolved from experiment of biomolecules, and usually this is taken from an online open source database such as the Protein Data Bank. And we take these structures of these interesting biological systems and we apply dynamics to these systems. So use the equations of motions to sample how these large and complex structures move around in solution. And from these sort of trajectories and movies that we capture of these large molecules moving around in solution usually, we want to understand biological questions of interest, for example, drug discovery, protein interactions, proteins in membranes, etc. And so what we do is we take these simulations and we will often analyze these to try to understand some biological questions. So we might look at the conformations that are changing in these systems and understand how, or the ensembles of how these conformations are changing. We might take these systems and look at where regions might be of interest of where a drug might be binding. And we want to also then be seeing how these large molecules interact with each other. And really, you can only do this with dynamics. There's also fields beyond the actual simulators running the simulations. People are now wanting to use our simulations for other fields of research, and also simulators are doing the same. So they're taking their systems and doing even more incredible science with them, such as training machine learning models to predict, new molecular conformations and interactions with better better force fields, for example, that are trained on quantum mechanics data. We might be also generating synthetic data, such as synthetic EM micrographs from simulations. And there we have a ground truth of what is actually in these micrographs. And we can use this to better interpret images that are produced from experiments. There's also looking at refining molecular ensembles that are sampled from simulations and refining these with experimental data, such as using NMR data to apply restraints to flexible molecules. Also, we will take our simulations and want to develop, test, and apply analysis methods to existing simulations, such as free energy calculations. And then also others may want to take our simulations and then apply the same protocols to their own systems of interests. The problem, especially with the last one, is when you want to reproduce or use simulations for your own systems of interest, you need to know how the simulations were performed. So here's an example of a standard simulation protocol that is usually published as part of a scientific manuscript. And we have a small space for where we can describe what we've actually done in our simulations. So in the pink is what was done. So we've taken a system, we might have minimized it, we might have changed the protonation states of our side chains, added solvent to our systems, and then also the tools that we use to do this. We will say, you know, what simulation packages we used, maybe the versions if we're lucky as well. But if you wanted to reproduce this, it's very difficult because we haven't described exactly how we've done and performed our simulation. So in the cases where we have seen an interesting paper and want to, you know, use the simulation protocol, we may be able to do this if we are part of a research group or a collaboration where these protocols are well established. So we can ask senior members in the group how they've performed these simulations and then try to replicate what they've done through their hand-holding. We might also look at online tutorials of simpler systems and try to adapt them to our more complex systems. We may also look at trial and error to try to reproduce published protocols from scratch. But this will be very difficult because we don't know exactly what parameters were used in each of the steps. We might have to have some educated guesses, and this may take a while then to try to replicate a protocol and try to see if we can actually get a simulation to be running. And lastly, we might even think to email the original authors and see if they have a nice simulation protocol that we can just use. And this might be more difficult to do. So another thing is, as well as knowing how to run the simulations, is where to find them or where to deposit your simulations. And we really have kind of two options currently available. The first is sort of large general purpose data repositories, such as the four listed here. However, these, because they are general, it's quite difficult to find where the simulations are submitted. So unless you had the final link to say, here's this simulation that's been submitted, it'd be very difficult to find because these large purpose data repositories lack the tooling to capture the metadata of describing the simulations. And so there's no way to really find them. There are smaller and field-specific databases which really give you data sets of maybe the same protocol for a simulation and then applied to different systems. And these are great. However, again, it's difficult to upload your own simulations to them, or how were these simulations performed. And also they may lack the ability to store large data trajectories because they have minimal data infrastructure available to do this. So with the cases for the large data repositories, if you did want to submit, then it's up to you to decide how much you want to contribute in terms of metadata and describing your simulations. So there is a lack of continuity in how simulation data is currently deposited. And also, if you do have a large system that you would like to submit, it's often very difficult because these larger purpose data repositories will have a limit. So if you have terabytes of data, you won't be able to submit to these. Now, other areas of biology are very good at sharing data, and we know this because when we perform our simulations, that's exactly where we're getting our structures from. And so it's really now we need to think about how can we provide this level of service as our other colleagues in biology do for biomolecular simulation. So this is kind of where PSDI and BioSim Data Resources comes in. So we are one of many partner projects as part of PSDI. And as James mentioned, PSDI is offering data infrastructure in physical sciences. And just with other support research communities like COSEC, our partner project is supporting the CCPBioSim and the HECBioSim communities, which is represented by 63 UK institutions with over 1,000 researchers. And so this is funded by EPSRC. We are here to try and sort of bridge that gap between the cool, community focused aspect of BioSim, so the training and the conferences that are provided, and then the code that is supported by COSEC and CCPBioSim, and then also the HPC focused software that is provided by HECBioSim. And so what's really missing is the data component, and this is where BioSim Data Resources is kind of trying to bridge that gap. So PSDI infrastructure as a whole and where BioSim sits in this is, so PSDI has a gateway which allows you to access all the services that are currently provided by PSDI. So this is not just BioSim specific, offer tools such as data format conversions, ways to capture workflows, machine learning tools, and then also ways to index data across various data resources. And so this common search is there to hopefully be able to link up many different data sources from many different themes of physical sciences. And so biomolecular simulation is one of them. So PSDI, the core partners are University of Southampton and STFC, and they sort of handle the grey parts of the infrastructure. And the collaborators are what are really fleshing out the blue and the green parts of the infrastructure. And so more collaborators are joining PSDI to try and develop more of these data services and data sources for PSDI. So onto the sort of main point of this talk, and that's the BioSim Data Resources component of PSDI. And there are four main components of how we are looking at data infrastructure for BioSim. And the first is the data provenance tools. Second is a data repository. Third is the metadata schema to describe the data. And then training resources for each of these three components. So first onto data provenance. So we use a lot of different software and tools to perform and set up our simulations. So where we perform our simulations, we may be in the cloud, we may be local, we may be using high performance computing. And then there's also the software and hardware that is used to run the code. And then the environments that the code is used from. So we might be virtualizing our software and then running from there. And then the final component, which is sort of the main thing for BioSim, which is the actual software programs that we use to set up, perform, and analyze our simulations. So there are just a wealth of different tools that we can use. And this is, I think, the strength of BioSim is that we have so many tools available to run simulations. And there's never one correct way to perform a simulation. We have many tools and they all do a particular job for a particular system of interest. And the idea is to try to incorporate all of these tools and not to try and sort of narrow ourselves in what sort of tools we support in capturing how these simulations were performed. So to sort of capture the diversity of all the tools that are used to set up and perform simulations, we went down the route of using IEDA to capture simulation data provenance. So we want to capture how a simulation is performed, the tools that are used, and how this whole sort of pipeline is working for a particular project and for this to be shareable with the community. So IEDA offers a Python infrastructure for workflowing. And what we have done is we've used IEDA to wrap simulation commands into the IEDA workflow engine. So I mentioned that we want to support as many pieces of software as possible. And we like that there is a diversity in tools that are being used. But we found that the simulation engines that we mainly use for biomolecular simulation are GROMACS and AMBER. So we've tailored these tools to work very well for these particular pieces of software, but they also work with any software that you run via the command line. So they're not just for GROMACS and AMBER, they're for any tool that you use that is performed, where the command is performed on the command line. So the idea with AIDA is to set up your environment for your software. So either a Python environment, install your AIDA plugin, and then ensure that your tools that you're using to capture your simulation provenance is available on the command line. And really what the user should be worrying about is basically just running their commands on the command line. That's all they need to do. And everything in this third column is abstracted away. And in this third column is how AIDA is working to try to capture the provenance of your simulation data, so how it's performed, the inputs and outputs for it, and then extracts any useful metadata. And all of this is not required by the user to see, and all they would need to do is run their commands as they would. And what is left at the end is the user can sort of package all of their provenance into a single file and then share this to a data repository for others to use. So this is practically how it looks like if you use one of these AIDA plugins. And in this example, this GROMACS command is all you would need to do to run our provenance tool. So it looks familiar. Users will know how to submit this command to the command line. So there's a low adoption to using this tool. And again, like I said, the complexity of using the tool is abstracted from the user. Now, if you did want to incorporate this sort of workflowing tool into your own sort of simulation automation, you could do and use the API. And this is Python-based, and you can then automate provenance capture within your tools as well. And what ends up being captured at the end is sort of graph representation of the data flow for each of the steps that you're performing. So you'd have the actual command that you use to run, so it's a process, the inputs for that particular command, such as the code that was used to run it and the files that were used as the input. And then you have an output of all of your files for that step that you performed. So in practice, if you were to run multiple commands, you would then see in the back, you would see this sort of graph representation of those inputs and outputs. And then finally, a full graph of all the connections for all of the steps that were performed in your simulation workflow. And this is then packaged and then can be shared. So we've decided to try and obviously make sure our tools work. So we've started, one of the case studies was to try and support one of the DRIIMB projects, which is looking at heterogeneity in cryo-EM data. And we've worked with Joel and Tom in CCPM to create a simulation workflow. So Joel and Tom don't perform simulations themselves, so we wanted to create a workflow that was automatable and then also capturable using our AIDA workflow tools. And so in this example, we wanted to sample the hinge of this small protein. And this workflow is capturing how we've set up and performed this simulation. So we've used AMBER tools to do the parameterization, PDB to PQR as a command line tool to protonate our protein, and then create the topology with AMBER tools, and then convert into GROMACS, and then run enhanced sampling with Plumed and GROMACS. And this whole workflow we've been able to capture with our provenance tools and then store as a single file. And so the idea is to hopefully share these workflows once this heterogeneity challenge is published and the results are published for it, so that all of the data that was produced to create these sort of synthetic micrographs for this challenge can be shared with all scientific communities. And what we hope to do is to expand the collection of workflows for other DRIIMB projects as well that integrate simulation with experimental data, which I'm sure will be talked about in further talks in this series. The second case study was looking at working with actual biomolecular simulators who know what they're doing. They have a workflow set up already for how they perform their simulations. And we wanted to see if we can aidify that and store the full provenance. So again, this is a slightly more complex example because not only do we need to take our structures, we need to remove low confidence regions, which you might usually do using a GUI or a visualization tool. So we've had to think about how do we kind of capture how we're changing the initial protein structure. So we might use a tool like SED to show how we've manipulated a PDB file to start off with. And then we use GROMACS tool to orientate the protein, and then Martinize to coarse grain the protein, and then also a modified Python script to embed the protein into the membrane, and then finally run the simulation in GROMACS. So each of these steps are captured with our tools, and we have tutorials available for how this workflow was developed and created. So in Docker for local use, and then we also have the tutorial in our documentation as well. Okay, so that was data provenance, and then we move on to actually sharing the data that is produced from simulation. And within PSDI, there is a component called data collections where various partner projects can create data repositories for the data in their particular field. So we've created BioSimDB within data collections, and the data collections is based upon Invenio RDM. So if you've ever used Zenodo, it's the same technology. So it's open source. It's well used. It's able to support something like Zenodo. So this is why it's been used in PSDI. So at the moment with BioSimDB, you can deposit your data if you are UK based. Any deposited data is available to all. Currently, there's no hard limit to the data size, and we have over 100 petabytes of high performance S3 array storage. So currently we have 2 petabytes, but this can be increased if needed. So we have the data available, or data storage available, to store simulation data. Now, another component that we are developing is an easy way to deposit simulation data. And one strength of using Invenio is it's a very general data repository tool. However, because it's quite general, we can't capture the full data or metadata from our simulations to the extent that we want to in terms of how we describe our simulations, all the inputs, the input parameters, for example, and output parameters as well. So we are going to incorporate data deposition directly into the CCPBioSim website, where we will have a very simple deposition form where all users would need to do is provide some minimal information such as contact details, citations, and title and abstract, and then the files that they would like to upload. So a topology and trajectory that is readable by MD analysis, so is what we require, and then a provenance tool, a provenance file, ideally using IEDA tools. And then we have a component where once these files are uploaded, we can automatically extract the simulation metadata from these files there and then. And the idea is for the user to be able to see what metadata was extractable from the files that were uploaded, and then see any fields where they can provide that information manually if required. But the main goal is to make this as automated as possible for extracting metadata. And if provenance files are provided, then all of this information can be extracted automatically. So with simulation metadata, there are a lot of ways to describe our inputs and outputs from our simulations, and the parameters that are used vary in terms of how they are described and what units are used for the various different software that is used. So one thing that we are keen to do is to have a representation of shared terms across various molecular dynamic engines. And we're working closely with colleagues at the Molecular Dynamics Data Bank to try and have a metadata schema that can be used across many different data repositories, so that when we are searching for simulation data, we are all using the same language. Now, in terms of how what happens to your simulation once it's been deposited to BioSimDB, we are aiming to go for a community-led data curation process. So once the simulation is uploaded and the simulation is then submitted for review. And the idea is first to ensure that the fields are filled in correctly, and this will initially be done with humans, so BioSimDB data curators, but we hope to make this process automatic. And then what we really need is we need the BioSim community to review the scientific relevance of simulations that are submitted. So it's not necessary that they need to be published in a high-end journal. It just needs to be scientifically significant, and that's where the community can tell us this information. And once that's done, so once there's some backwards and forwards with the reviewer and the depositor, the record can then be published via SimDB and accessible. So as final slides to just say kind of what our vision is for data production and consumption in the biomolecular simulation field. So apologies. So we imagine that users will be able to create simulation workflows where we know exactly how a simulation was performed, and so therefore is more reliable to be used. So the final data then is more reliable to be used in other areas of research. So have the workflow of how a simulation was performed, and then upload that to a data repository, so share it with others. So either that could be on, just any general online repository, or the BioSimDB, where if that is the case, if something is uploaded to BioSimDB, then we can extract the metadata to describe the simulation. Users will be able to download workflow blueprints, rerun simulations, and modify simulation workflows, and then be able to run their own commands, and then again upload their new data to BioSimDB. And we're keen to ensure we make connections with data repositories that are also being developed by MDDB. So they are doing an amazing job in creating data repositories where we have amazing analysis and visualization of simulation data, and we want to ensure that our tools are compatible with the tools that they're developing there. And so the idea really is to have a collection of high quality simulation workflows. And if we have a repository of these workflows, we can see what tools are most popularly used in BioSim. And if we know what tools are used more popularly, then we also know how to improve these tools, or develop new tools that may improve simulation workflows. And then also it makes our data more accessible to other researchers in other fields. So having our data available to the wider community, it enables many different things. So I mentioned the workflows, being able to have a full record of how simulations were produced. We'll have somewhere to store our data and share it with other communities, and be able to develop more tools compatible with the next and current generation of HPCs. We'll be able to reduce duplication of effort. It will be greener because we can reuse simulations because they're deposited and available freely, and be able to share data or knowledge through our community more easily. And then again, this enables other fields such as AI and also new analysis methods of our simulations to be developed. So with that, I'd like to thank you all for listening and thank our collaborators and developers on this project.
Cool, thank you. So Sarah, you're handling the questions, aren't you?
I'm handling the questions, and it was a fabulous talk, Jas. So Pete, there are lots of questions. I think you sort of answered this, but maybe you could expand on it a little bit. So James, hi James, lovely to see you, has asked, what is the collaboration with MDDB?
So at the moment, it's not extremely official, I suppose. But at the moment, what we are doing is we are working to develop a metadata schema that describes simulation data. So what we're hoping to do is to have the same language for how we describe our input parameters, our output parameters from simulations, and then be able to populate our databases with this. So if we were to search for these terms across databases, you should be able to find the data through either BioSimDB or MDDB or any other data repository that uses this schema. So that's what we're currently doing. But maybe James wants to expand upon other ways we could work or are working with MDDB.
Yeah, so I mean it's early days, but so we went to their user meeting, which were the CCAM one in Italy, and we've been discussing ways to collaborate on infrastructure. So it's early days, but we've agreed that we are going to do this. We're working with the Oxford node in the UK, and we're going to look at how to deploy MDDB infrastructure alongside BioSimDB infrastructure, and start to link this curation process together and feed data down into their pipelines. But like I said, it's obviously there's a significant technical journey with that. We've had the source code for about a week. So yeah, it's still very early days, but we're sorting that out in the background.
Brilliant, thank you. Said Eduardo asks, could you use Ada to capture processing in a GUI written in Python?
Potentially, yes. So there are GUI tools available in IEDA called like IEDA Lab. But you can also, especially because there is an API, you could develop a GUI on top of that. The issue is with simulation protocols is we don't have a fixed workflow. So our workflow will change depending on the system that we're interested in studying. And the tools that we use are so varied that a single workflow usually doesn't do the trick. But if you have a particular protocol that will always be the same for the systems that you're studying, then that's where a GUI is probably quite convenient because your workflow is not changing, so your structure is fixed. But with what we do, these workflows are constantly varied, and so what we're trying to do is capture the various tools that we're using. And then what you could do is you could potentially take one of these workflows and say, hey, I'll make a GUI out of it. But that's not something that we are doing.
So 2 questions from James. So he asks, if we have already run simulations in GROMACS, can we capture the data provenance afterwards with Ada?
No, it's the short answer. So what you could do is you could rerun like your setup steps. Hopefully that shouldn't take long to do. So that at least you've got the protocol down. And obviously your simulation bit, you don't have to rerun that because you've got your final simulation all done. But you would need to rerun your steps to then capture that into your AIDA workflow.
And can we use other data provenance capture formats like JSON files from Cyprian and Workflow Hub?
Yes.
Excellent, excellent. That's really cool. So I'm just trying to make sure that we haven't missed anyone. So Samuel asks, so one potential approach to utilize large shared resources such as Zenodo is to build overlay types of data banks. So they've provided some links to lipid bilayer simulations to make us all aware, and said this provides currently metadata and programmatic access to hundreds of MD simulations. On the other hand, MDverse, and they've provided a link, can be used to search MD simulation data from Zenodo. I just wondered if you wanted to comment on the use of Zenodo and how we interface with those resources.
Yeah, definitely. So one good thing is like the repository that we are using is built on the same infrastructure as Zenodo. So if we can upload our data to something that is like Zenodo, then these tools, for example, the APIs that we create and the sort of domain-specific metadata that we populate, if you want to upload to Zenodo, you can do, and then you can use our tools to search for them. I guess the issue is from the box, Zenodo doesn't provide you with the ability to store your domain-specific metadata. You'd have to create a community to do this. So we've basically created that community, and then we'll give a framework for how metadata should be described for this community. And then there's no reason why this couldn't be used then for other data banks. And yeah, MDverse is very cool in what they're doing, and they are searching by looking at like file formats or looking if GROMACS was used and just from the file extensions, and that's really cool to do. But then you still are missing that information of what were the parameters used to run these simulations. It's usually just the output files that are stored, and you're missing then the how of how it was performed. And hopefully what we're doing will enable that. I hope that answers your question.
Cool. And Mark, hi Mark, asks, have you considered using BioSim space to aid with or at least encode workflows and file conversion? So for example, PARMED does not always faithfully convert topology files between AMBER and GROMACS.
Yeah, so if BioSim space is, if you can run it, like, I guess obviously you can run it as a script or on the command line, then yeah, it can. The good thing about these workflow tools is you can easily incorporate any software that is sort of anything that's automatable can be incorporated into these workflows. So it's more of a question of it doesn't matter. What we're trying to do is say it doesn't matter what tool you want to use. We just want a way to capture how you use that tool, and this kind of enables that. So yeah, definitely, this could be one of the sort of workflow steps for converting between two formats.
Cool. And Zahir asks, can we use these resources to run MD simulations?
No. So yeah, unfortunately, it's for storing the data. But potentially, I mean, there may be something in PSDI where you may be able to at least use some data resources to maybe set up simulations. But I think running them, which requires a lot of compute, you'd need access to HPCs or a compute cluster to do that.
Maybe in the future we can have an order form for simulations. That would be nice, wouldn't it? So Sam asks, are there or will there be integration with common pre-processing steps like TLEAP to capture how researchers generate their input files? For example, I often find that I hop between TLEAP and H++ to determine protonation states, which I think could be a useful step for other researchers to be aware of.
Yeah, that sounds really cool. So like especially when there's something like quite finicky about how you actually do the protonation, and if you're flicking between tools, but you can capture that with AIDA, and then you can publish this workflow to say, look, this is how I do this particular component of my setup, and even if it's just a small, it's an important step. It's not the whole workflow, but this particular way is how I do it. And if you, as long as it's not like a web server, as long as it's something that's runnable as a program, then we can capture that and then share that workflow. And it would be so useful to actually have those sorts of resources on BioSimDB.
So Anonymous asks, does this workflow capture interactive command line inputs such as termini selection in PDB to GMX?
It does, yes. So this is one of the reasons why we focused on Amber and GROMACS, because there are some commands that require interactive um, options by the user. So we do make sure these work for those. There is also, if you have a tool outside of these that also uses interactive commands, there are ways to capture it, but it's just a bit more involved to do. But it is possible.
Excellent, thank you. And James has got two more questions. Can we share existing Zenodo repository links so we don't have to upload them again?
James is saying yes, so then yes.
Excellent, that's cool. And this question we've talked about before. How can we capture charm GUI steps in our ADA workflows?
Yeah, so we have spoken to the Charm GUI developers, and this is something that they do want to do. They do want to capture provenance. Now, we can't do it with our current tools because it's obviously you do this online. But we're kind of happy to talk to them about either incorporating AIDA, but it sounds like they do want to maybe create their own workflow tools to capture, how you've done your Charm GUI steps. And if that's the case, the output of that can be shared then, or we can try to link that with our AIDA tools.
So I have a question, if that's all right, and I'm wondering if other people are thinking the same thing as me, but maybe are a little bit more shy than I am, so don't want to ask. But when I'm setting up my simulation, I make lots of embarrassing mistakes. And I don't want to share all of the stupid things that I did internationally when I was trying to set up my bit of DNA or my protein. So when I'm using IADA to capture my workflow, what do I do if there are mistakes? How do I deal with that?
If the mistake is like part of your workflow, then keep it. But if it's something that you can easily delete nodes from it. So you can try running a step and it didn't work, and then you can just remove that node and try again. So it's not like it's set in stone of, you've run this step and that's it. We're going to see your mistake forever. That's definitely not how it would work. But I guess there is some curation involved in like how I would use it is I would kind of develop a workflow or develop a protocol that I know that works, and then I'd IEDify it. So you can make all your mistakes that you want, but then once you have something that's working, that's when you want to then capture it and then share that, is how I would do it. But if you want to share your mistakes, that's also encouraged.
I suppose, but because one thing that it would be good to do is have like a private IADA and a public IADA. So like I would do my own thing with my with and make all of the mistakes, and then and then I can go through and check that the perfect way of doing it works, because it is quite, going back and redoing something.
Mm.
I mean, that's the whole point of the provenance capture, isn't it? Is it's not always that easy to remember what you did.
Yeah. And like it is all private, it's all local anyway to begin with. And it's only when you package it and then you decide to share it that it's available. So it's not like everything you do is recorded and stored in some secret server that we can watch and see what you've done wrong. It's all private, and then you can then just share your workflow at the end.
That's brilliant. Thanks very much. And another question that I think the community will really want the answer to is, when is BioSimDB available to receive trajectories, and how do people actually start giving data to the project?
I mean, so it's live now. I don't know, James, like, is there a particular what's the word? Is there a way that we want this to be done, or is it kind of, let's just get people to upload?
Yeah, okay. So we're in early alpha release, right? So that means that we actively want the community to come and try these things out. So at the moment, we're going to be very careful about who's using it, and kind of like, we'll be sort of controlling access so that we can get information about what's working, what's not, this kind of thing. If you're interested in taking part in this early alpha access, I mean, I will say it's probably quite a complete alpha. I mean, it probably is almost very production ready, right? So we're just sort of, PSDI itself as an infrastructure is in alpha, so naturally we're in alpha with it, right? So PSDI is probably going to move to phase 2, which is what we're going to call the production phase. At least that's what they're calling it with UKRI, in sort of April time next year. So until then, we'll be really sort of asking people to just come and kick the tires, try uploading things, tell us what's slow, what's fast, what works, what doesn't. We're not going to cry if you tell us something's, rubbish, just come and tell us, come and try it. So best thing to do is to email me or Jas, and we'll take it from there with the alpha phase.
So James Krieger's helpfully posted it in the chat. We can access it. James says he can access it. Well done, James. You heard it here first.
I mean, I don't know what happens if you click that new upload button, because you will need a UK federated account to actually do that. But it's I'm surprised it's the upload button's there without being.
Yeah, I think you can like upload, but then it won't be available on the repository yet. So it would have to go through like the review process, and then that's probably where it gets stuck at the moment.
Okay, so that's, yeah, okay. So best thing to do is to go on BioSimDB itself, is to come and talk to us. And then we'll guide you through that process.
And what is the email address that you want to be contacted on for queries about BioSimDB?
I'll just post the email addresses in the chat. At the moment, it's just us. So we will be setting up some general email addresses soon, but we've not got those yet. I'll just post these in the chat. There you go.
Oh. Okay, brilliant. And Fatima, I can see your question, but I don't think we know the answer to your question, unfortunately. So Fatima's asked about how we get a protein structure from powdered X-ray diffraction, and I just don't know the answer to that, I'm afraid. But maybe somebody in the on the webinar does and can help you. Okay, so this is really, really exciting. So thank you so much, Jas, for an amazing talk. You've had lots of appreciation in the chat that I haven't been able to feed to you. So hopefully this is the beginning of something really, really exciting. And there will be another DRIIMB online talk in the new year, and it's going to be Helen Berman talking about community building and how you get everybody to work together, which is, I'm so excited. It's going to be amazing. So thank you very much, everybody. I think we're at the end for today. So we'll see everybody in the new year.
Yeah. Thank you all. Thank you all
Thanks,
for joining us
Mariana.
today.
Bye.
Thank you.
Bye.
Great to see
Bye. Bye.
everybody. See you soon. Bye-bye.